Welcome to the Oprah Winfrey Show. Today we will be speaking with Dr. Patelis and Dr. Lazarus, both from the Department of Natural Resource Sciences in McGill University. Welcome.
Thank you for having us. Yes, it's quite the dream.
Now, I heard that you have been looking into the ecological niche when it comes to competition and the environment. And this study was done with the use of seabirds?
Yes, so we wanted to dive deeper into the analyzation of the ability of seabirds of multi-species to coexist in the same locations, especially due to the competitive displacement principle.
Could you explain to us what that is?
Of course. So the competitive displacement principle tells us that competitors aren't able to successfully coexist and survive if they share the same ecological niche, such as habitats and resources. And because of this, niche differentiation arises. Yes. Niche differentiation is when competitors partition or divide their resources and habitats for survival. And this division allows competing species to coexist in the same niche with limited overlap for resources and habitats. And we wanted to look into how seabirds in these colonies adapted to this differentiation. For example, we wanted to know if this increases high interspecific competition.
Why did you choose seabirds?
We found so interesting about seabirds is that they form island colonies with a variety of populations of species that actually end up overlapping in their foraging and diet. And we wanted to see how they're able to coexist in this environment with overlapping ecological lifestyles and how this affected their interspecific competition.
Oh, okay. And which seabirds did you decide to study and investigate?
We collected data from 3 ox species: the Atlantic puffin, common murres, razorbills, and the layered species, the black-legged kittiwake.
Ooh, and which specific islands are you focusing on?
We focused on 2 islands called Calculot des Bertrands and Isle de la Maison, both located in the Gulf of St. Lawrence in eastern Canada.
Ooh, okay. Well, now that we know the focus of your experiment, how exactly did you execute it? What were your methods and materials?
We had numerous materials and methods in this experiment, including the GPS tracking, diet sampling of ox species, spatial analysis, and diet analysis.
Ooh, okay. Tell me more about the GPS tracking.
Why, of course, we attached GPS loggers onto the lower back feathers of birds, or on the 4 central rectrices. The use of these GPS loggers were to track birds when returning to their nest. They would record latitude and longitude every 5 and 15 minutes, respectively.
Oh, and how are you to ensure that you can recapture these birds when they leave? Did you have some nesting area for them?
We actually installed wooden nest boxes near breeding grounds that automatically closed when they were captured. And so we captured the murres with a leg hook or with our hands. We captured the puffins by hand. And we captured the kittiwakes using mist nets, noose mats, hand nets, and noose pulls.
That's brilliant. Now tell me more about the diet sampling of the ox species. I heard that you had to investigate the differences in their diet, correct?
Yes, that is correct.
Oh, okay. And how come this investigation did not include the kittiwake?
Oh, well, fun fact. They actually regurgitate their prey rather than carrying it with their bills, so we were not able to get accurate information.
I see. Now let's discuss the spatial analysis. So you didn't include the puffins nor El de la Maison, right?
Yes, no, they were not included, and this is because of insufficient sample sizes and lack of data collected due to GPS malfunction.
Oh, how inconceivable. But how did you identify the foraging behavior of the other birds? Correct me if I'm wrong. You categorize each behavior as being at the colony, flying, or foraging.
You are correct. In fact, we computed different foraging trip metrics. We looked into the maximum distance from the colony, trip duration, foraging range index, the distance from when they made their first stop, the total number of food patches visited, and the time they departed to forage.
Oh, could you explain to me what you mean by foraging range index?
Why, of course. So the foraging range index is what we use to get an estimate of the average distance from the colony that the birds are foraging from.
Ah, I see. So let's say that the foraging range is high. Does that indicate the bird travels farther from the colony to forage?
Precisely. We compared what prey each bird species will mostly go for, the ability of the prey and the sizes of the prey. For example, from our collection of data, both the Atlantic puffin and the common murre frequently forage on capelin, while the razorbill would go for sandlands. In fact, we noticed that each of these birds forage on a different prey species or even a different size suiting their beak. This can be seen in Table 2.
Okay, could we pull up their other data table?
We also noticed that when it comes to the distance and time of the bird's foraging range, it shows that the black-legged kittiwake and the common murres have a longer travel time than the razorbill. The black-legged kittiwake even has a higher FRI than the common murres and razorbills, which is indicated on Figure 1.
Now let's pull up an image of Figure 1. So, enlighten me, what was the main goal you're trying to research? What did you hypothesize before conducting the experiment?
Throughout the experiment, we mainly focused on the question, does niche differentiation in foraging habitats among seabird species have an effect on high interspecific competition?
And what was your hypothesis?
We hypothesized that due to the high amount of competition, the multi-species seabirds adapted different evolutionary traits that allowed them to coexist with limited overlap.
So what did your results reveal?
Well, as you notice in Data 1, there was very low overlap due to differing traits. For example, murre's wings have 30% higher loading, which allows them to forage deeper and farther than other species, like the razorbill. And as you see in Figure 2, murres are flying farther and faster due to their size.
Fascinating. This relates to what you said earlier about the niche differentiation?
Precisely. Because of the difference in traits between the murre and razorbill, they forage in different locations, diminishing interspecific competition. They're able to coexist without constantly competing for the same resources and killing each other.
Oh, okay. Now, would you say this type of coexisting has evolved over the years?
Of course, this evolution is precisely the reason these differing species have evolutionized with the traits they have. Their diets are segregated, and therefore so are their foraging and breeding locations. It's all a matter of surviving.
Surviving in the colonies.
Yes, which is what makes it all the more fascinating. These habit differentiations are a major consequence of interspecific competition, and play a big part in colonial living for the seabirds.
Well, your findings were quite fascinating. I'm very intrigued with the results you had from the study. It's truly interesting to learn more about the connection the different species have with the seabirds and their choice of prey. Now, I do have some follow-up questions.
Ah yes, go on. We would be happy to answer any questions.
Now, I'm curious, if these birds were to breed and reproduce over time, let's say about 10 years from now, which birds would most likely be able to survive the longest?
We think that the murre will most the longest. If you remember from our data results, the murre had the largest amount of land where it foraged and sought both food and shelter. They had an advantage and an expansion of shelter and resources, and they had a high foraging distance and duration, and most likely to evolve, which makes them most likely to evolve and adapt for survival.
And I would assume that their beaks would also change in shape and size as well?
Definitely. Throughout the years, the ecological niche these species live in will change, affecting the species' foraging habits and lifestyles. Because of this, competing species will need to adapt by evolving their traits, like the beaks, shapes, and sizes. They will probably need to change in order to adapt to the new food and eating habitats that will result due to the evolving environment.
And would it be possible that there are environmental factors that may have affected the travel distance, foraging, or even the diet of the birds? Like, was there a change in temperature, weather patterns, or surplus or scarcity of food that contributed to the behavior and characteristics of these birds?
Yes, definitely. We cannot control nature, and while we manage to maintain an overall idea of the foraging habits based on the species traits, the overlap in niche is not entirely limited and led to foraging for the same resources in the same locations on several occasions.
Well, there you have it, folks. Again, let's give an applause to our special guest for today. Thank you for sharing all of this information.
Thank you for having us. Bye.
Bye.